So without further ado, I'm Kelsey. Obviously, you all know me, but I do marketing for Pureline, and I'm out of Watertown. And our group here is sales and product development mainly, but we're spread all over the place throughout Washington, the Midwest, and Colorado. And with me today is Ellie Boda. She's our research director. She manages the team out in Warden, Washington for the research farm, and she leads the way in different projects for research and development at Pureline. So excited to share with you guys all about, what we talked about. So before I send it over to Ellie, we're just going to talk briefly about what we do at Pureline. Let me get this. All right. So a little history. Pureline was established in 1948, and we were originally a pea and bean breeding company. We're established in Moscow, Idaho, so that was the origination where all of our growers were. Pretty much everything was in the Idaho area except for the research farm. And it wasn't until the 1970s when the research and productions facilities moved all into Washington State. So we've been there since, and we've been family owned and operated for the past 75 years. So we just recently celebrated our 75th anniversary, which was a really exciting opportunity for our team. And we've been family owned and operated. It wasn't the same family throughout our journey at Pureline, but our most recent owners purchased the company in 2019. That was Ethan and Tori Rosine, and they live and work out of Warden, Washington as well. We have different facilities throughout the U.S., but our main two production facilities and growing regions are in Warden, Washington, and recently Olathe, Colorado. So pretty much everything, the heart of Pureline is located on the Pacific Northwest in Warden. So from, harvesting, planting the seed to milling, cleaning, everything in between is done in the production facility in Warden. And just down the road from there is the research farm. And that's where we do all of our selections, all of our different research projects and our trial fields. And just everything is, as far as research goes, is done at the research farm there. And we also have a couple location in Minnesota as well. We just purchased a facility in Colorado, Olathe, Colorado in 2023. So we moved some of our productions that we did in Washington to the area of Colorado, but we also were able to expand into some different crops that we weren't producing before. So some wet seeds such as watermelon and cucumber. And then we also do some pumpkin, sweet corn, and squash. So that's been kind of an exciting opportunity for Pureline to expand into Colorado. Just to explain a little bit more about, our markets and where our products go and how we sell. We sell mainly peas and beans, and that's been kind of our mainstay since Pureline was founded. And we recently expanded into edamame. So we're in the 4th generation of edamame, so nothing commercial yet, but that's been an exciting expansion for Pureline into a new crop segment. So we sell primarily to processing, so this would be canning companies, frozen foods such as Birds Eye, Gerber Foods, those types of companies. And we sell, all over the U.S., into the Midwest and Pacific Northwest, pretty much all over. And then we also ship internationally for the processing market. In 2007, we expanded into the fresh market. So this would be anything that can be consumed fresh. So in the retail side, anything, the home garden segment, anything like that. We expanded into crops such as tomatoes, broccoli, peppers, anything kind of like that. And we're not particularly breeding these varieties, but what we did is we partner with suppliers in other countries that don't have representation here in the U.S., and we brought those products over to the U.S. market. We trialed it extensively in Wisconsin, and we still do that today. To see which products fit best within the marketplace. Then we sell to a dealer market who then goes and distributes through those channels. Then the last area that we sell into for our product segment is the dwarf varieties. So this has been a newer thing for Pureline the last 7 years. we partnered with a company in the Netherlands called Prudak, and they breed and develop these small, compact tomatoes and peppers and brassicas that can be grown indoors and are bred for low light situations. So this is for things like people who don't have a large garden or don't have any space in like urban areas can grow produce. So that's been kind of an interesting project for Pureline as well. So the last thing before I hand it over to Ellie is just want to talk through the four main emphasis at Pureline. And so I've already talked a little bit about our seed production, and obviously that's a large part of what we do at Pureline in our different locations in Washington and Colorado. But within our seed productions, we always try to use, different technologies to keep up with the growing trends within the market and produce, the highest quality seed we can. The second thing is our seed health. We're continuing to produce a healthy seed crop year after year for our growers so that they can have the best harvest and yield possible. So within our process, we take a lot of preventative measures to ensure this as well. And then the last thing, we also provide certain varieties for certified organic productions. Third is our strong relationships. So relationships are pretty crucial for Pureline, and from the beginning, this has been the most notable aspect at Pureline is just our relationships between, our team. We have a tight-knit team, even though we're, spread out throughout the US and into Canada and all over. We have a pretty tight-knit team, and communication is key in that, but also our relationships with our suppliers and customers. And these are all built on trust and reliability to make sure they're getting the best service possible and the best customer experience as possible as well. And last but not least is our plant breeding. So I'm not going to go too far into this as Ellie's going to take over here, but just to give you a brief introduction, we use induced variation and make a selection to provide the best and most productive products out there. We also use non-GMO practices to produce, the resilient varieties for the field. And so Ellie's going to take over now, and she'll explain a little bit more about our processes and our best innovations coming through the pipeline.
All right, good morning, team. I'm very happy to see my amazing research team all here. So thank you for doing that. We are in a busy planting season. But Kelsey and I were talking about, giving this training to growers and technicians and agronomists in Colorado. And then we said, our team should also benefit from it. So thank you for that. Congratulations, Kelsey. It's her birthday and she's working hard with us. So if you want to send her a message later, would be nice. So let's talk what we have been doing, and it's good for us as a team to recap and for people that are watching this video as a training in the future. I started in breeding over 15 years ago, following the agronomist, the senior breeder, and identifying varieties, all the hard field work that breeders do, and they were established. But from 1950s on, there is a shift in plant breeding. Breeding is an ancient technique. As soon as humans establish communities, they start selecting and choosing and pollinating. So it's a very old technique. But in 1950s, we have the technology of DNA, decoding our genetic codes and using that in our benefit to conform or to configure how we're gonna select plants. So what we do is we produce organic and sustainable seed using marker-assisted selection, and it's always non-GMO. So if everybody outside Pureline will ever wonder what we do, we take advantage of markers, the DNA knowledge, to flag, like farmers flag their fields, we flag our genome of the crops we work on. We work with 3 crops. They are legumes from the Fabaceae family, so they are self-pollinated. They have capability of connect with bacteria from the soil, rhizobia, and other microorganisms, and fix nitrogen right from the air. So it's very important, and we like our crops. So they are peas, beans, and edamame, which is the green soybean. So next, please. So this previous work in breeding was established in 1800s by a guy called Mendel, Gregor Mendel, and he was a gardener, like our technician Nando, walking in the field and observing with a very good eye. So he started working with peas and he started calculating what's the inheritance or how many of those traits are passed from the father or the mother to the offspring to the next generation and so on. So he followed it for 7 generations until these lines were pure or were kind of almost homozygous. And he came up with theories. So this is called qualitative genetics. Let's see, I have a mother that has a good Fusarium wilt resistance. Fusarium wilt is a discrete trait. So Mendelian trait is either yes or no or 1 or 2. Either the mother passed to the son or not. And it's simple inheritance or genetic from Mendel. Next, please, Kelsey. So here's an example. We discussed that with many times in our team. So if I have a round pea versus a wrinkle pea, or a yellow pea versus a green pea, or a purple flower versus a white flower, these are the things Mendel observed. And he discovered that if he crossed, let's say, a yellow seed with a green seed, the first generation, the babies that we collect, they're going to be homogeneous. They're going to all look the same, like all yellow. But if he planted again, the grandbabies will have segregation within the same pod. That photo was taken in our fields, and you can see there is yellow seeded peas and a green seeded pea in the same pod. So it's a very cool observation, and you can calculate the ratio or which traits are dominant from the mom or dominant from the dad. So that's very basically. And again, we go from choosing the father and the mother and planting 7 generations to make it homozygous. Next, please. But again, with the advent of DNA or the discovery of DNA, the genetic code, people start worrying about complex traits. So Mendel worked with pair of genes. So half of what we inherited from the mother, half from the father, the yellow seed, the green seed, or maybe 2 or 3 traits combined. But people start observing that another Fusarium disease, Fusarium root rot, that's Fusarium solani, it's not like the mother is resistant, the father is susceptible, and then you cross, you can have some yes or no, yes resistant or no, not resistant. It's complex. It varies in a continuous, and we call it quantitative genetics. Yield is a trait like that. Height, if you measure everybody in our group, everybody stand up and we measure our height, we will see that there is not short and tall. There is a whole continuous of variation. Maria is a little taller than me, then Steph's a little taller than Maria, and Nando, and so on and forth. So we kind of a bell curve trait. So this is complex traits that we do need DNA markers. Again, yield. In humans, if you want to think obesity as well, diabetes, some traits that have different expression. Next, please. Let's look at pinto beans or the cranberry beans that Nando works. It's very similar to that. So if you get some pinto beans in the same bag and you look at a handful of each, you're gonna see that they have stripes of brown, but the stripes are not exactly the same. Some are a little higher, some have more, some have less, some are almost white, some are darker. So there is a continuous variation of that. So what I can see on a plant or on an organism, I call the phenotype. So anything related to phenotype, phenome, or things that are observable, or the Mendel's work before the genetics, with the DNA assistance, we call the phenotypic variance, or as Kelsey mentioned before, we look for variance. So we create that variance through breeding, mom and dad, and have generations to follow, or the innovations that we are implementing now. But all this variation is mapped in the genetic code of the organism. So if I bring these pinto beans to a lab and I want to look at the stripes, I will gonna realize that everything that's in the seed that I can observe is also linked to the gene or the genotype. So the collection of genes or everything that I map with DNA is called genotype. So phenotype, observable with my eyes. Genotype is the genes, the DNA, the RNA, the things that we're gonna see in the lab. So if you look at that, how does this DNA look like? We code it for us humans to understand better. So it's a whole bunch, four actually, pair of nitrogenase bases, and they go from GATCG or variations of that form. Sometimes every time when a plant is growing or our body, just as we talk right now, there's cell division. Our cells are dividing. The DNA is in the nucleus of the cell. And it's copying the sequence from one cell to the next cell. And there are errors because it's a lot of codes and it's a lot of common frequent errors. Some of those errors or variations called mutations. Some of these errors don't yield on anything. Every 3 pair of bases can code for amino acids that will code for a protein. So that genotype will become or be translated into phenotype or things that I can observe, like in the pintostripes. But just no need to be worried, just for you to have an idea how the DNA and the phenotype relate. Next, please. And how do I see the sequences? Okay, I take a leaf tissue or a drop of blood or anything, and I bring to a lab to be sequenced. So they are a spectrum of light, so chemical molecules that as they pass through the light, they have different intensities. That's how I know those bases are placed in the way they are. But to confirm, so I observe some variation on my pinto bean or the cranberry bean that Nando works, and I collect a little bit of the tissue, leaf tissue or the seed coat, and I send to the lab. So what we do, we powder with liquid nitrogen, for example, and amplify the DNA, and then we run in gels, electrophoresis, that will show the lights in different bands, just like in forensic files, or if you want to know a paternity test. So some bands will look like the father, some because we are half from the father, half from the mother. So we have different bands in our DNA. And after the lab confirmation in that gel, we can check our plants in the field again and continue improvement. We can select the best that have that trait and also look for agronomics. Next, please. From the 1800s to now, people were really focused on breeding for the seed quality and the aerial quality. But when you are in a field, people did wonderful things. Our previous breeders at Pure Line, they did everything they could at the top part of the plant, or maybe before 1950s, we didn't have all this technology. But there is one area of the plant that we can improve because there's room for us to work on. It's the roots. The roots of the plant, you can see, especially in our legumes, the 3 crops we work on, they form little houses, nodules, in which they host the friendly bacteria from the soil, the rhizobia, and they pull together the nitrogen from the air and they pump up the plant. So yay, sustainability. So we are applying less fertilizers in the future because of that. So legumes don't need as much fertilization. And then through this root breeding, we investigate as well. We're doing greenhouse studies, and sometimes we look if there's other pathogens. So yes, the legumes make a house, and the bacteria is in the soil, so there's a communication. Some of our growers inoculate, so there is not enough bacteria in the soil, rhizobia, because sometimes over irrigating or the pH is too low or too high. This particular rhizobia, they need a pH that is very touchy. So if it's too low or too high, it won't grow. So we won't have in our soil for the next season. That sometimes make us inoculate as well. Next, please. And yield is king. So our breeding program is always focused mainly on yield and all the yield components, right? The quality of the seed as well, the quality of the pod that we are selling, because we sell green vegetables. And we also want to make sure it has all the resistance and the stresses tolerance, like drought, for example, that we need before we release to a farmer. But if you look at yield, for example, there's a whole bunch of components that play a role in how much a plant will yield, like stress. If it's drought or if it's still humid, this plant will have a lower yield. There's genetic factors that are inherited, like how many pods per plant or how many berries per pod. That's genetic inherited. And then also there's a hormonal crosstalk. So every plant, like a human, needs hormone, and they have different hormones. Gibberellic acid is one that we are treating the seed with, and it will help to give that initial boost on growth. We will talk more about hormones in a minute, but those are compounds produced by the plant in order to regulate growth and life stages. From a baby will have different hormones that mature plant or than us, and then as we get older, the hormonals shift again. And then external physiological factors, if there is like a bug or an infestation or light intensity, photosynthetic efficiency. Here I want to talk about a little bit about edamame, because we have this crop grown in Washington state, but our light intensity per day is not as efficient as in, say, in Texas. So we can make this crop work, but we'll thrive better there. That's an external factor playing a role. Nitrogen use efficiency is not enough to put nitrogen in the soil. Each different genetics of each one of our varieties uptake the nitrogen in a different way. So it's interesting too to see those variations. We can even talk about breeding for nitrogen in the future. But for now, we're gonna be good like what we are. And water. One of the main focus back to the quantitative trait, the newer genetics, is, for example, Fusarium root rot that I mentioned before. Now I'm going to guide you through this case study that we are doing in our farm. These are different varieties of peas, and some have different levels of resistance to Fusarium root rot solani. It's not like one have, okay, it's total resistance. That's how we call sometimes intermediate resistance or highly resistant or moderate, partial. It depends, but goes in a scale from 1 to 5. Generally, 1 is very clean, like you see on the left, and you have some that the roots rot on the right. That's very susceptible. Could be Bolero, because no secret that Bolero doesn't have that tolerance. But look at these plants for a minute. If you plant in the field and you never check the roots, the top or the aerial part is fine. So by the time the grower realizes that something wrong, start firing up from the bottom of the plant up, is too late. So the whole plant will be dead and we'll have a loss up to 50% in the fields. Next, please. As far as we are concerned, and research staff did some plating with me, and Nando and Maria do now. Everybody participates, Leslie and Bo also supervises. So we can do from the plants or from the field. How this is look, a black ring around the plant. If I cut that root in half, you can see it. It's plugging the root system from getting water and nutrients. If I plate it, I can see that nice round colony shape. Next, please. And if I bring it to the microscope, you can see little canoes or little organisms like that one in blue. That's the characteristics of the Fusarium root rot or solani. Next, please. I can also incubate. We can collect this disease from the soil or the plant. We just finish a test from the soil. And in that way, we wake up the pathogen with a selective media on an agar plate, and then we transfer for Kerr's media, which is a liquid media, to wake it up. And then we can access, that's okay, Kelsey, you can go next. And then you can access the growth indicators. We always treat things inoculated and on a control after a soak. You can see that there is variation on 2 varieties in the blue tags in the left. You have some that can survive the disease. Look at the roots, it's still the black ring, but it's still with roots and can survive and yield. So it's a moderate resistant or high resistant. And Bolero over there rot the whole seed. It was totally sensitive. And then you can look in our controls, just make sure we are doing a clean test. That was just water soak. Next, please. Kelsey? Okay. And we do this in a controlled environment. We do like for a growth chamber. So when we are doing inoculants, we don't place in the greenhouse, or we try not to place anything in the soil afterwards. So we have an amazing engineer there, Bo, and Nando came up with that. And it seems simple, but it saves a lot of time because you are inoculating these plants, a lot of them inside of a controlled chamber that you can clean and also keep the inoculant from spreading around in the farm. So next, please. This is a picture of the chamber inside, and we got awarded in among the 10 top research in breeding around the world. So good job in the conference that we attended last time. So we have the test, and we have a humidifier inside to keep the moisture. We tested beans for white mold, and we tested peas for mildew, downy mildew. Next, please. Another focus of breeding and research is drought. We have been discussing that, and there is changes in temperature in different growing areas around the world, and this is very important to focus on. So we do greenhouse tests that Leslie and Lupe are starting today. We place those seeds in a very hot plates, let's say in our greenhouse now before going to the field where it's very hot. And we also control water so we can have influence on drought. So heat and drought to test together. And we send to different areas like Georgia, and other companies report to us that we have materials that already have heat tolerance like PL0008. So that good job, team. We are on the right track for future generations to come. Next, please. So another picture of PL0008. This was taken in New Zealand, Australia, I believe, both areas there, and they have a nice quality straight pod bean. So we're going to continue to check if our standards are there. Disease resistance, we introgress, and we check with the lab for markers. Next, please. So this was reporting some of our beans, and just another emphasis on our beans. Next, please. And the newest technology that we rescue from 1950s when they first invented DNA is mutation breeding. And I'm gonna take a little bit of time to talk to you guys about it. It's very important. So it's non-GMO. We are not taking the DNA and from one species and transferring to the other. So this is a dip or a radiation that can occur, or natural variation on the DNA. Instead of having naturally, we induce it. Next, please. So if you look at the mutation breeding, so again, every time a cell divides, it copies the DNA. Sometimes there are errors. And if we can't repair, we have within the DNA a repair system, a whole bunch of enzymes that go there, find the error, cut, and paste. Very cool, very clever. Every time as we talk, this is happening. The same with the plants. Sometimes, if this mutation cannot be repaired, it becomes a tumor. And people, like observant people with a good breeding eye, or in the field and see that sometimes in the soil, plants have naturally tumors in them. And trees have tumors, the black balls that you see in there. They start investigating and they find that there's an organism in the soil called Agrobacteria tumefensis that naturally infects plants and cause tumors. So some of that they're using for technology, and especially the GMO team, they love that bacteria because they can induce tumor. But in this case was a chemical dip. So not in our lab, we outsource for a lab that's safe to do it. They got a strong chemical, they take our seeds and they dump it in a chemical for 5 minutes, no more than that. Then they washed and that's all done. All the genes or those mutations are shifted. So we use EMS as our protocol. And when the seeds arrive to us, they are all clean and ready to go, but they are also full of mutations. And this mutation is not specific. is random. So from dipping 4,000 seeds, sometimes we ended up with 5 plants. That's success. And from those 5 plants, we plant it again and we select for the traits we want. It can be done for any trait, from herbicide resistance, as the agronomist was mentioning in our last talk, or it can be for yield and quality traits. What does mutation do? It speeds up. Remember, Steph, and when we discussed, yeah, we have a mom and a dad and a pea, we cross, and then we have to plant 1 generation, 2 generations, 3. It might take 7 years or more before you have a homozygous line. And as far as mutation goes, you can have the dip, plant 1 more time, and check for variation should be stable. So this will save us a lot of time down the road. Next, please. And now with this, I want to ask if you have any questions, and make sure you can contact us afterwards, and we are happy to discuss any other things that you might be curious about. Thank you very much. Anybody has questions or?
I actually have one quick, Ellie.
Yes.
I know with the PL008, would you see a lot of varieties coming out of the beans that we're doing more for the heat tolerance? I know there's other factors that you're breeding for at the moment, but is that a pretty high priority for beans, is heat tolerance?
Excellent question. Heat tolerance is important in certain areas in the US. Beans have a high genetic by environment interaction, which means if I plant them in Washington and breed them in Washington, when I send them away, they might not have the same traits that I select for in Washington. So we are sending to trials in Florida, South Carolina, and focus on heat, and it will help a lot of other areas in the world. But we also focus on salinity. Maria is starting another test in the germinator, because the waters from irrigation over time build up salt, and you want to make sure you have varieties that withstand that environmental challenge. So we do 2 main areas here. We do like resistant to biotic stress, bio, life, every disease you can imagine. And we do resistance or tolerance to abiotic stresses, which are drought, salinity, heat. Since 2008, we invested a lot of time and thinking. Our team worked very hard to innovate or to make the step into the molecular or the DNA breeding, or to have this marker-assisted breeding to help us to be competitive, but friendly competitive nowadays to bigger corporations. And we have a lot of lines in the pipeline coming forward.
It's exciting.
I don't know if Nando, Steph want to add anything. Bo, Maria, Lupe, Leslie. If not, we can thank you very much for your participation. And if you are watching this video later, just please contact us under this comment under the link, and we'll be in touch with you.